biological carbon pump of the world's oceans is one of Earth's most powerful climate-regulating systems. This natural mechanism is a vital component of the global carbon cycle. It consists of biological and physical processes that transfer carbon from the ocean's surface, where it is captured by microscopic organisms, to its deep layers, where it can remain stored for decades to centuries. By doing so, the biological carbon pump plays a central role in reducing atmospheric carbon dioxide and regulating Earth's climate. At the heart of this process are phytoplankton, tiny, photosynthetic organisms that inhabit the sunlit upper ocean. These organisms absorb dissolved carbon dioxide from seawater during photosynthesis, converting it into organic carbon. Some of this carbon is recycled near the surface, but a significant portion begins a journey downward, carried by sinking particles, ocean mixing, or even swimming marine animals. While scientists have long recognized the importance of this carbon pump, large uncertainties remain. Key questions persist about how much carbon is exported from the surface, how it is transported to the deep ocean, and how long it remains sequestered once it arrives. In response to these questions, the 2022 study by Nowicki et al., Quantifying the Carbon Export and Sequestration Pathways of the Ocean's Biological Carbon Pump, employs a global modeling approach that integrates satellite data with oceanographic observations. Their aim is to quantify the contributions of the primary pathways through which carbon is exported and ultimately sequestered in the deep ocean. These include the passive sinking of organic particles, physical mixing processes, and the active migration of zooplankton. By resolving these pathways at global and regional scales, the study provides a clearer picture of how ocean life helps stabilize our climate and sets the stage for understanding how this balance may shift in a warming world. To investigate the ocean's biological carbon pump, Nowicki and colleagues developed a comprehensive modeling framework that simulates how carbon moves through marine ecosystems and is ultimately stored in the deep ocean. At the core of their approach is a global, steady-state ocean model that couples biological, chemical, and physical processes. This model simulates a simplified food web in the upper ocean, capturing interactions between small and large phytoplankton, different classes of zooplankton, including those that migrate vertically, and the production and transformation of organic matter. The focus is on two primary forms of organic carbon, particulate and dissolved. These are tracked as they are created in the surface ocean and transported to depth. The model is driven by satellite-derived estimates of ocean productivity, which indicate how much carbon is fixed by phytoplankton through photosynthesis. These satellite data also provide information on the size structure of the phytoplankton community and the depth of the sunlit or euphotic zone. This allows the model to simulate how much carbon is generated and which organisms are responsible for its transformation and export. Physical transport of organic material is handled using an ocean circulation model based on real-world tracer data, such as temperature and salinity. This ensures that the model accurately reflects how ocean currents move carbon throughout the water column. To improve reliability, the model is calibrated against a wide range of observational datasets, including dissolved oxygen, organic carbon concentrations, and biomass estimates of plankton. By adjusting model parameters within an ensemble of 124 different configurations, the researchers captured uncertainties in ocean dynamics and biological behavior, ensuring their conclusions are robust across plausible scenarios. This integrated approach allows for a detailed, data-informed simulation of the key pathways through which carbon is exported from the surface ocean and stored in the deep sea. The study's model reveals a global biological carbon export from the surface ocean to the deeper layers of approximately 10.2 billion metric tons of carbon per year. This export is primarily driven by three key pathways: the sinking of organic particles, the active transport of carbon by migrating marine animals, and the vertical mixing of dissolved organic matter. The most dominant of these is the gravitational pathway, which accounts for around 70% of the total carbon export. This process involves the downward flux of carbon-rich particles such as phytoplankton aggregates and fecal pellets produced by zooplankton. Of the gravitational pathway, fecal pellets are responsible for the largest share, about 6.2 billion metric tons per year, while aggregates contribute around 1.1 billion metric tons annually. The second major pathway is the migration of zooplankton. These animals feed in surface waters and then descend to depth, where they respire or excrete carbon. This active transport mechanism contributes approximately 1.0 billion metric tons of carbon per year to the deep ocean. The third contributor, the mixing of dissolved organic matter by physical processes, adds about 1.9 billion metric tons of carbon annually to deeper waters. While less efficient than particle-based pathways, this mixing pump still plays a significant role in certain ocean regions, especially in subtropical gyres. Together, these processes not only move carbon into the ocean interior, but also determine how long it remains sequestered. The model estimates a total carbon storage in the ocean interior, resulting from these processes, of roughly 1,300 billion metric tons. The average time that this carbon remains stored varies by pathway. Carbon transported via gravitational sinking remains sequestered for about 140 years on average. The active transport by migrating animals leads to a slightly longer storage time of roughly 150 years. In contrast, carbon mixed downward in dissolved form is typically sequestered for only about 50 years due to its shallower penetration and quicker recycling. A key insight from the study is the strong spatial variability in both carbon export and sequestration efficiency across ocean regions. High-latitude areas, particularly the North Atlantic and North Pacific, emerge as the most effective zones for long-term carbon storage. These regions exhibit high rates of export, primarily via sinking particles and migrating zooplankton, and benefit from deep ocean circulation and colder temperatures. These factors promote the deep penetration of organic carbon and slow its degradation, resulting in average sequestration times that often exceed 150 years. The Southern Ocean also contributes substantially, though limited observational data in polar regions introduces some uncertainty. In contrast, the subtropical gyres display lower carbon export rates and significantly shorter sequestration times, averaging around 50 years. Here, the dominant export mechanism is the downward mixing of dissolved organic matter, which tends to remain in shallower waters and is more readily recycled. These regions are characterized by warmer temperatures, more stratified water columns, and a prevalence of small phytoplankton, all of which reduce the efficiency of the biological carbon pump. The study's biome-level analysis confirms that the spatial pattern of carbon export and storage is tightly coupled to ecological structure and ocean circulation, with high-latitude systems acting as the planet's most effective long-term carbon reservoirs. Overall, this study offers a comprehensive and integrated assessment of how the ocean's biological processes contribute to the long-term removal and storage of carbon from the atmosphere. By modeling the three primary transport pathways, sinking particles, migrating organisms, and dissolved organic matter mixed downward, the researchers have clarified how these mechanisms interact to shape the global efficiency of the biological carbon pump. One of the most important takeaways is that these pathways are not equally effective. They differ not only in the amount of carbon they export, but also in how deeply and how long that carbon remains stored. This highlights the importance of considering both export magnitude and sequestration timescales when evaluating the ocean's role in the global carbon budget. However, several uncertainties remain. The spatial distribution of migrating organisms, particularly in undersampled regions like the deep ocean and polar seas, is still poorly constrained. Additionally, the model assumes fixed behavior and metabolic rates for key species, which may not reflect real-world variability under changing environmental conditions. These limitations underscore the need for better observational data and refined biological representations in future modeling efforts. Looking ahead, Understanding how climate change may alter the structure of marine ecosystems, stratify ocean layers, or shift dominant carbon transport pathways is essential. As ocean warming and acidification progress, the efficiency of the biological carbon pump may decline, reducing the ocean's capacity to buffer atmospheric carbon dioxide. Studies like this one are critical for building a mechanistic understanding of Earth's natural climate regulation systems, highlighting the complex interplay of biological and physical processes that govern current ocean carbon storage is essential for predicting how the global carbon cycle might evolve under climate change.